All right. Hi, everyone. Welcome back. We are now in the last week of the course, and today we have a lot of ground to cover. So I will be going through in greater detail on each of the major study designs. So today I'll cover randomized control trials and two of the most important observational studies, cohort and case control. And then on Thursday, I will cover quickly cross-sectional surveys, diagnostic studies, and ecologic studies. And then Taryn will give the talk on meta-analysis, which will close the course. By the end of the course, you will all receive the quiz, which is the post-course quiz, just for you to go through and see what progress you've made in your learning. And then we will also send you a link for course evaluation, which you will receive from Rosalind at Becky Cisa, for you to give us some feedback on how you like the course, what worked well, what didn't work well, and how we could potentially improve and do better if we were to do a similar course in future. So I will now share my screen and get started with the lecture on the major study designs. Can you folks see my slides?
Yes, you can see them.
Yes, yes, we can.
Okay, great. All right. So if we go back to one of the earlier lectures where I had given you like a flow chart on various study designs, basically we think of all epidemiological studies in two really broad buckets. One we call experimental research, and the other bucket is called observational research. Randomized control trials is a very important example of an experimental research, right? Every randomized trial is a human experiment. We are doing things that are not naturally occurring. We as investigators are intervening. We are giving the medicine, we are giving the vaccine, we are doing a strategy that normally may not have happened. And then we want to see by intervening, we are able to change what might happen in terms of outcomes and so on and so forth. In observational studies, we don't intervene, we don't give the medicine, we don't give a vaccine. We merely look and see what happens to people who may have already taken a vaccine, who may have already taken a drug, who may be using a condom, may not be using a condom. It is endless, who may be smoking, may not be smoking, but we don't tell who to smoke, who to wear a condom, so on and so forth, right? We merely observe and we either follow them over time or do other measurements to see what their natural outcomes are. So that's the biggest difference between experimental research and observational research. At its simplest, a randomized control trial is one where individuals who are participants in a trial are allocated at random to receive one of several interventions. At a minimum two, sometimes we even do trials with just one arm. That could be an early phase 1 type of a trial. But usually it's a 2-arm parallel design that is the dominant design within randomized control trials. Is it possible to have 3 arms? Yes. Is it possible to have 4 arms? Yes. So long as you have sufficient numbers of people, there's adequate power in the trial to tell the difference between the groups, then the answer is yes. And as I mentioned, randomized control trials are human experiments. The intervention, whatever the intervention is assigned by the investigators. Patients never get to pick what therapy they want in a randomized control trial, okay? The minute patients pick, then all bets are off. It's no longer a real trial because patients decide to take something or not based on their perception of risk, based on other things, comorbid factors, and many other things that are beyond the control of the investigator. And the minute people are picking on their own, we are now in an observational realm. We're no longer in an experimental situation. Treating doctors, whoever is treating the doctors, they don't get to pick. They can't tell the investigator, you know, I like my patient, put him in him or her in the drug arm and not the placebo arm. No such things are allowed. Okay. neither the patient, nor the treating doctor, nor the treating nurse, nobody gets to decide who gets what, right? Protecting that is fundamental in a randomized controlled trial. Randomized controlled trial are usually comparative. So there's a control, the controlled in a controlled RCT. The C in a RCT means randomized controlled trial. Control means there is a control arm. We can compare what happens to the hydroxychloroquine group against the group that got something else, right? Sometimes uncontrolled trials are done where there is only one arm, and that is the treatment arm. And usually a phase 1 trial is an uncontrolled trial, okay? Now, here are all examples, high profile examples, not the only ones, of randomized controlled trials on COVID-19 treatment, okay? So this is a randomized controlled trial on preventing COVID by using hydroxychloroquine. This is a trial on lopinavir, ritonavir for people with severe COVID, remdesivir for the treatment of COVID, dexamethasone for hospitalized patients with COVID. And these trials have been extremely influential and helpful in deciding which treatments work or not work in this pandemic. By now, we kind of know already that hydroxychloroquine is finished, right? There is no future for this treatment. Lopinavir, ritonavir is finished. There's no promise for that. Remdesivir is promising. It's not a blockbuster drug, but it does seem to reduce the amount of time people spend in the hospital. But by no measure is this a magic or a miracle cure for COVID-19. Dexamethasone, again, dexamethasone has got nothing to do with the virus. It's more to do with the inflammatory response or the hyperactive host immunity, which seems to calm down the hyperactive inflammatory response, and it seems to benefit people with severe COVID-19. That seems to be a useful therapy in some people. This is just a snapshot of the evolving literature on COVID treatment. And honestly, 2 months ago, none of these existed. So we would not have really known at that time whether any of these medicines work or they are totally useless or harmful. And that is why randomized trials are so critical. And the best time to do a randomized trial is when we are starting to think about a potential treatment, right? Very early on, when we really don't know whether a medicine will work, not work, or even be harmful, that is the right time to justify a randomized controlled trial. But if we already started using a medicine and it's already widespread in clinical practice, it becomes so much challenging at that point to do a randomized controlled trial, right? Too many people are already on a treatment. But we must, if we don't do randomized controlled trials, and if all we had was observational studies on hydroxychloroquine, remdesivir, or dexamethasone, we would still be scratching our heads on whether these medicines work or whether it's just bias in those observational studies which make them look as if they're working, right? By doing a randomized controlled trial, and if they're done well, and if there are enough subjects, we pretty much can bury a research question for life. And that's why randomized controlled trials are absolutely fundamental for things like drugs and vaccines, right? So why do we need a randomized controlled trial? No other design can get us close to that counterfactual ideal comparison that we spoke about when we talked about causality. To say something is causally linked to something, we need a group that is exposed to that cause, and we need the same group to be unexposed at the same time, and we want to know what happens to the two groups, right? In that case, everything else is constant. The only thing that is different is that one group got the medicine or vaccine, the other one didn't, right? One was exposed, the other was not. We know that is impossible, but a randomized controlled trial gets us close to that ideal comparison where we can say, we're not worried about all other factors. The only thing that is different between these two arms is one got the drug, the other didn't get the drug, right? That ideal is simulated by a randomized controlled trial. It's not perfect. Randomized controlled trials can also get it wrong, That randomized control trials can also be biased, as you will see in a second, but a well-done randomized control trial that has adequate number of people, adequate power, can really, really nail a question and then settle the controversy. Okay, but randomized control trials are not for everything, right? There's a whole bunch of research questions that are simply not amenable to experimentation, right? Does tobacco smoking increase the risk of lung cancer? Well, nobody could have done a randomized trial on that question, right? Does drinking alcohol during pregnancy increase the risk of fetal alcohol syndrome? You can't randomize someone to drink or not, right? There are lots of questions in daily life that are completely outside of the purview of randomized control trials. That's why they are perfect and ideal almost for certain domains. Like for treatment domain, randomized control trial is practically God, right? Treatment, vaccines, if you don't do randomized control trial, you will never really have the evidence to decide whether a vaccine or a drug works. But for harmful exposures, it's very, very hard to even think of a randomized control trial. And many exposures are harmful, right? Whether it's tobacco smoking, or whether it's diet, or whether it's physical activity, sexual behavior, you can't really randomize people to any of those. But sometimes people can be randomized even in those situations. Like we can't randomize someone to smoke, but we could randomize someone to smoking cessation to see whether the risk of lung diseases comes down over time, right? We can do that. Diet looks very difficult, but there have been really good randomized control trials on certain types of diets to see whether certain diets are more protective than others. So since randomized control trials are human experiments, we will always get informed consent. It has to be done after ethics approval. Protecting individuals is paramount. In other words, we will never sacrifice individuals for the sake of a randomized trial, right? Individuals come first. And that is a critical fundamental tenet in terms of ethics. We cannot randomize people to harmful exposures, and we cannot randomize people to a medicine and then a placebo if there is already an active treatment available, right? Whatever is the standard care for that particular disease must be the comparison group. Occasionally, we can pull off a placebo when there is absolutely no current treatment available. That's why some of these COVID trials have been able to use a placebo, right? We don't have a vaccine for COVID right now. So it would be acceptable to have a placebo vaccine, and there are placebo vaccine trials happening right now for COVID, right? Like the Moderna vaccine from America just started trials today with something like 3,000 or 30,000 people. And the placebo, they're using the Moderna COVID vaccine in one arm randomly, and the second arm is saltwater injections, right? That's saline injection as placebo. So it is justifiable because there is no vaccine for COVID right now, right? Could you do a placebo-controlled trial on an influenza vaccine? The answer is no, because there's already an influenza vaccine available, even if it's not perfect. But if you have a really better vaccine, then you can have the better vaccine as a treatment arm and the existing vaccine as a comparison group, not a placebo, okay? Placebo is a no-no if there's already a vaccine or a treatment available for a certain condition. Lots of ethical issues. Unfortunately, we don't have time in this course to cover those sorts of issues. So what's random allocation? Random allocation means that all participants have a defined probability of being assigned to a particular arm, okay? And the allocation, who gets what, that's what allocation means, who gets what, should never be determined by the investigator. Let's say I am the clinical trialist. I should have no power to pick and choose, you get this, you get that. That's not my job, right? I will not have any control over who gets what. The treating doctors never have control over who gets what, and the patients will never have any say in who gets what. So if I were to volunteer in a COVID vaccine trial, I cannot go there and go to them and say, beg them to give me the vaccine and not the placebo. I'll have zero say in the matter. All I will know is that I'm getting a shot, and the shots will look exactly the same, right? I have no idea if it's saline or if it's the actual Moderna vaccine or an Oxford vaccine. I will only know much later what I got or didn't get, okay? That is true random allocation. And that's also blinding, by the way, right? Blinding is automatically inbuilt in it, right? Because it's the exact same looking vaccine, I have no idea. I'm blinded to what I got. So I'll explain blinding later on. And allocation should not be predictable. For example, if you use an alternative scheme, right? First patient gets drug, second patient gets placebo, then again drug, and then placebo, and then drug and placebo. Before you know, the entire hospital has come to know that's the pattern, right? And then people will jockey around and move themselves up and down the list to make sure they get into this or make sure they get into that. The nurse will move patients around. The treating doctor will mess around. Before you know, the entire trial is a disaster because it's a predictable pattern. That's why Mondays, Wednesdays, and Thursdays, or you come on this day, I'll allocate you to treatment. None of that are good methods. They're all predictable and they're dangerous, right? Nobody should have the whiff of who's going to get what, and protecting that is absolutely paramount, right? So what does randomization do? In real world, your decision as a person or a patient, my decision as a patient or a doctor, we don't make random decisions in real life, right? In real life, if I am taking metformin, I am taking metformin now, okay? I'm taking metformin every day because I'm pre-diabetic, because my dad died of diabetes, right? So I'm worried. I'm doing something to prevent diabetes. It is tailored to my risk profile, okay? I'm a little overweight. My dad died of heart disease. He died prematurely. So I'm thinking all of that. And then that is what led me to that point of getting onto metformin. So if I took metformin, you cannot separate all of those other characteristics that I have. I'm a male. I'm about to turn 50. All of that goes along with that decision to be on metformin. Why did my doctor write me a metformin? Because she knows all of this story. She didn't make that decision randomly, right? Real world is never random, right? Real world is you tailor the treatment to somebody's risk profile, right? And so then if you compare all the people who are taking metformin to those who are not in a natural database where it's not randomized, you never know whether the differences you're seeing are because of metformin or because of other characteristics that led people to take metformin, right? And you already saw that when I spoke about hormone replacement therapy. Which woman decided to take hormone replacement therapy? She was younger, she was richer, she was thinner, she was white. All of that led her to have better outcomes against heart disease. It wasn't really the hormones in the end, right? You cannot disentangle a woman's decision to take HRT or stay on HRT from all her other traits. The only way you can snip that relationship between covariates or comorbidities or other risk factors and the drug is to assign people randomly, right? If I'm randomly given metformin, then the link between metformin and my profile and my father's ill health and my family history is all gone, right? Then you get people who are eligible, you randomly decide this gets treatment, that gets placebo, and then you look at the table, compare the treatment group and the placebo group, which is what every single trial will show you. You will see beautiful balance of age, sex, smoking, family history, obesity. I don't care what you look at. Known and unknown confounders will be balanced between the treatment arm and the control arm because randomization guarantees that, right? In a real world study, if you compare treated group with the untreated group, they will differ on 50 variables because that's what decided them to be on treatment or not on treatment. You cannot snip the relationship between the risk profile and the treatment. Randomization is the only way we know in epidemiology where we can guarantee that somebody getting on a vaccine has got nothing to do with their risk or other risk factors. It is a true random event. And that's how an experimental design is infinitely superior to an observational study when it comes to medicines and vaccines. So take a look at the remdesivir RCT, right? Remdesivir, 500 people, placebo, 500 plus people, right? Two arms. Then this allocation was done randomly. And you look at age, right? Age group, 58 years in the treatment group, 59 years in the placebo group. Males, 65% remdesivir group, 64% placebo group, right? Asians, 77%, 57%. Slight difference here. But otherwise, you find pretty much in every other variable, there is virtually no difference between the two groups. And that is what randomization produces. Occasionally, you can have imbalance, even though it's random, between one or two covariates. And if that happens, we can always adjust for that in the analysis. But a randomized control trial is the only one that can produce beautiful balance in various confounders and take away the link between the treatment and the risk profile. So I told you already, nobody should have any say in who gets what. And protecting that is absolutely fundamental. And that issue is called concealment of allocation. Concealment of allocation means the person who's actually deciding to give the vaccine shot, right? Whoever that it is, research nurse, whoever that may be, that person should never have any say on who gets what, okay? So here is an example of how people can subvert randomization or random concealment of allocation. So this is a famous example. They did a surgical trial, right? And people were randomly assigned to either open laparoscopic appendectomy, which means you remove the appendix using a laparoscope, right? Minimally invasive, or you do an open appendectomy, which means you open the abdomen. It's a major surgery. Then you remove the appendix. The trial apparently ran perfectly during the day, right? But at night, it was much harder for the junior surgeons to bring a laparoscopic senior surgeon from home at night, right? If it's open, they could have just done it themselves. It's not rocket science to do it. But if the allocation came back as laparoscopic, they would have to call the surgeon, wake them up at night and bring them to the hospital and whatnot. So the junior surgeons started holding the envelope under light to see whether it said open or laparoscopic, right? They lifted the envelope up, right, like this, just to see what the code was, right? If it said open through the envelope, through the light, they would just do it on their own, junior surgeons. If it said laparoscopic, they will put the envelope down, take the next envelope, and wait until they found an envelope which said open, and then they would do the surgery, right? This is absolute no, right? Because if the sickness of the people who come at night is very different from the sickness levels of people who come during the day, and if the operating ability of the junior surgeon is very different from the senior laparoscopic surgeon, you've introduced all sorts of biases in this example, right? So this is a no, right? So there are techniques that are acceptable in terms of how we do the concealment of allocation, and there are techniques that are unacceptable, right? So for example, translucent envelopes, right, that you can see through, no. Allocating to different days, odd and even schemes, right? None of them are considered acceptable ways of allocation. The best techniques would be sequentially numbered opaque sealed opaque envelopes, sequentially numbered. So you know if people have been skipping certain numbers. There's also something called central randomization. If you have an eligible patient, you lift the phone, you call a call center, you give all the information, somebody at the other line says, okay, take the box with this code number and give that shot. You have no idea what the code number means. Could be drug, could be placebo. You have zero control over it, right? So there are methods which are bulletproof compared to the regular methods, weaker methods in terms of randomization. So let's take a look at one of the hydroxychloroquine trials to see how they did concealment or allocation. They had equal numbers of cards, equal numbers of hydroxychloroquine against placebo, okay? And with each group assignment number randomly generated by the computer, and were placed in sequentially numbered envelopes that were opened as the patients were enrolled. Okay, so I'm reasonably happy with this. I would have liked to have seen one additional word: sequentially numbered opaque envelopes, right? Opaque or thick envelopes that you cannot see through. That would have made me happy that these guys did a decent job. So trials come in very different names and flavors. It can be quite confusing. This is a standard FDA type of classification: phase 1, 2, 3, 4 trials. Phase 1 trials are usually very small, 10, 20 patients. They're not really to look at whether the drug is good or not. It's just to look for safety. It's just to look for side effects. And it's usually no comparison group, right? Usually you just start off. Before this, you would have done animal trials, by the way, right? You would not introduce a new drug or a vaccine without animal research first. Once you're in humans, start off with phase 1, right? Phase 2, a slightly larger group, maybe now you're reaching up to 100, and to see whether there's some improvement in viral levels or blood pressure or whatever you're looking for, and to evaluate its safety. May or may not have a comparison group. Phase 3 is a real 2-arm parallel group randomized control trials, right? Large enough, you would be looking at both safety and effectiveness, and it's usually good enough to make a policy decision on whether to move ahead and recommend the drug or not. Phase 4 trials are post-marketing surveillance, right? Once a vaccine is out there in the real world, rare side effects become obvious, right? So phase 4 trials are more about post-marketing surveillance to see whether a drug is safe. Some drugs have been pulled out of the market years after they were introduced because we came to know much later that they were quite harmful in the end, right? So depending on how participants are exposed, we can have parallel trials, we can have crossover trials, and there are many, many more complex trial designs, which I will not talk about. There are trials called factorial trials. There are trials called adaptive designs, sequential designs, non-inferiority trials, equivalence trials. So many different cluster randomized trials. It's a very large field, the field of RCT. So I'm just making it simple for you folks to understand in a short lecture. So in a typical parallel 2-arm RCT, take the Moderna vaccine trial, for example. They will have volunteers who agree to participate and consent, and then they'll randomly allocate to the vaccine group and the placebo group. Placebo group in this case is salt water, right? The person doing the randomization or allocation will not have any control on over who gets what. The patients who volunteer will not know whether they got the Moderna vaccine or the placebo. And then we'll have to wait, right? Why do we have to wait? Well, we have to wait to see what the outcome or the incidence rate of COVID-19 will be in the Moderna vaccine group against the saltwater placebo group. If we find a much, much lower incidence in the vaccine group compared to a higher incidence in the saltwater placebo group, that means the vaccine is effective, right? How long do you have to wait? Well, it depends on the number of COVID cases you're likely to see, right? They would have done that kind of math. They said, if we wait for 3 months, we are likely to see this many cases in this group, this many cases in that group, based on the hypothesis that they have, right? That's how they decide on the duration. That's how they decide on the numbers of people who are randomized to the treatment or the placebo group, right? This is typically how a 2-arm parallel RCT is done. Sometimes we have a crossover, right? We start off with people in the placebo group or a treatment group. Then we wait. We do something called a washout period. Washout means the drug is metabolized and removed from the body, right? Then we switch people. The people who were given placebo, maybe in the first week, then end up with treatment in the subsequent period. And those who started with the treatment then are given placebo. So the same person has an opportunity to be their own control, right? So 2 weeks ago, I took the medicine, and then 2 weeks later, I'm taking placebo. I can then compare how well I did on some metric while I was on the medicine, while I was on the placebo. So this is a more powerful design, and it's even closer to the counterfactual comparison that we care about, right? Because each person behaving as their own control is absolute gold, because all else is constant. Same genes, same family history, same risk profile, everything is constant. The only difference is that the time period is not the same, right? I cannot be taking the medicine, not taking the medicine at the same time, but I can take the medicine this week and not take the medicine next week, right? So time is a potential confounder here, but otherwise this design is even closer to the counterfactual gold standard design that we talk about. Right, blinding is another way we can cross-classify randomized trials. So you would have read, this is a randomized, controlled, double-blind trial, right? So they're saying randomized, then they're saying controlled, that means there's a control arm. Then they're saying double-blind, triple-blind, single-blind, who is blinded and how many groups are blinded, controlled trial, right? So there's a loaded phrase, right? So blinding means, like I said, several people could know who's getting what. The patients themselves, right? I could immediately, if I found out that I got the Moderna vaccine, as opposed to I got the saltwater vaccine, right? Now, why is that a bad idea if I came to know about it? Well, if then you ask me, so do you have any side effects, right? If I knew I got the Moderna vaccine, I might tell you, yeah, I have a lot of pain here. I'm having some slight fever. I have a lot of itching here, right? Our brain starts concocting all sorts of symptoms, even if it wasn't really there. The power of suggestion, right? But if I knew all I got was salt water, and then you ask me, do you have any adverse events? I say, I'm perfectly fine, right? So our reporting of soft outcomes can completely change depending on our perception of what we got, which is why a patient should never, ever know what they got, right? It influences their symptom reporting or their outcome reporting. Investigators should not know. The guys who came up with the Moderna vaccine absolutely should not be deciding who gets what. Otherwise, they might deliberately give the vaccine to healthier, younger people because they want to show that the vaccine works, right? No, can't be done. The doctors treating should never know, right? Because then, let's say my doctor knew I was on salt water, not on the active treatment, then my doctor, she might push me to be more careful about myself. She might tell me, you know what, you really didn't get the drug, you got the placebo, so you're still at risk of getting COVID. Put on your mask, don't go out, right? Then that messes with my incidence of getting COVID-19, right? That's a no, right? People do all sorts of things. If I knew you were on the placebo, I could give you other medications trying to overcompensate for the fact that you probably didn't get the active medicine, right? All sorts of crazy things are done in the real world that you can't have happening in a trial. Data analysts, I'm the one crunching the numbers, and then I say, oh, okay, I really want to see this drug work, right? Because it's my baby. And then I can use a different statistical model, right? I can use a different p-value cutoff to make sure this medicine looks like it's working, right? So even a data analyst or a statistician can be blinded to which group is which until the blinding is removed at the very end, right? In an open label trial, everybody knows who's getting what. There's no blinding at all, right? If it's medicine against a surgery, that's the comparison, then it's usually an open label trial. It's impossible to know it's impossible to blind in a surgical trial situation, right? Okay, so that's basically it. Single blind usually means patients are blinded. Double blind means patients are blinded, and usually those who are doing the data collection, the investigators, are blinded. And triple or quadruple blinding means other groups are also blinded, like biostatistician could be blinded and so on and so forth. It is up to the trial paper to clearly delineate who was blinded or not blinded, okay? So I just put together this cartoon by looking at the hydroxychloroquine trial, the remdesivir trial, the lopinavir, ritonavir trial, right? To see who was blinded, right? So in the hydroxychloroquine trial, nobody was really blinded, right? On the other hand, in the remdesivir trial, patients were blinded and the care providers were blinded. Lopinavir, ritonavir trial, none of the groups were blinded, right? So blinding is an important issue. A blinded trial is a much superior trial than an open label trial because the bias in outcome collection, data collection and reporting is quite acute if you don't do the blinding, okay? And I've given you a table showing what are all the possible positive effects of blinding, right? From the patient perspective, blinding makes sure that I do not over-report or under-report based on what I think I got, right? For the trial investigator, I don't probe you more because I know you are on this medicine or that medicine. I collect the data the same way, whether you are on treatment or whether you're on placebo. And I already gave you one example last class. In this aspirin trial, people were testing their own capsule, right? They were tasting their own capsule, and they came to know that somebody was on aspirin or not on aspirin, right? Because aspirin has a certain taste. Same thing, if you chew on hydroxychloroquine, it has a very peculiar taste. Some medicines immediately tell you that you are on it or not, right? The urine color changes, right? So sometimes you really cannot blind. If you take rifampicin, a TB medicine, your urine turns orange, right? So you cannot really blind someone in that situation. And generally, if people taste something and they come to know which medicine they are on, then they change the way they report the outcomes of the data, and that ends up creating a bias. Or if I thought I was on aspirin, but I tasted it and it just was a sugar pill, then I could go buy aspirin from the pharmacy and start taking it myself because I think I'm losing out by not being on aspirin. That's called contamination. You allocated someone to aspirin or placebo, but they ended up getting something else, right? That's called contamination of the two arms of the trial. All sorts of crazy things can happen if you don't protect against that, right? So concealment of allocation is at the time of randomization, the person deciding treatment versus placebo should have zero control of subverting it, right? They should not be able to subvert that process. Blinding comes after. Once you've started the trial, then you make sure patients, people collecting the outcome data, statisticians, etc., are not aware of who got what. Both could be done. But of the two, concealment of allocation is more important, should always be done. Blinding may or may not be done depending on the nature of the trial, right? Like I said, surgical trials, how are you going to blind? You can't really blind in that situation. So concealment of allocation is always possible. Blinding may be possible, may not be possible, depending on the situation. So bias, we've already discussed it last class. You can have severe bias if concealment of allocation is not done correctly, right? If I start cherry picking who gets the vaccine, who gets the placebo, then I will cherry pick healthier, younger, potentially richer people and put them in the vaccine arm. Then the other group gets the vaccine. The placebo is people who might be poorer, people who might be non-white, people who might be sicker, people might be older, and then boom, I see a huge difference between the vaccine group and the placebo group, right? And that may be nothing to do with the vaccine. It may be to do with who got the vaccine in the first place or who got placebo in the first place. So the only way to prevent people from cherry picking and creating the difference between the trial is to make sure nobody has a say in who gets what. That is the only way to make sure you don't have bad selection bias in randomized controlled trials, right? Information bias, I think the aspirin trial where people taste and then report outcomes differently based on what they know, That's an example of information bias. People collecting data from patients should also not know who got what. Otherwise, they can differentially ask more questions or probe more in one group compared to the other group, right? So if I knew you got a vaccine, I could spend more time with you probing about side effects. And if I knew you got saltwater placebo, I just ask one question and I walk away, right? Because I don't think you should have side effects, right? That's differential data collection. That's a no-no in a trial. So we need to make sure there's randomization, and then we can also make sure blinding is done during outcome ascertainment, right? So blinding is one way to make sure the integrity of the trial is maintained and nobody knows what they got, and the people checking on the outcomes don't know who got what. And last quick comment about power and sample size, right? How do you decide on a size of a trial, right? Is 100 patients enough? Is 200 patients enough? Well, it all depends on what the hypothesis is, right? There will probably never be a 100 patient COVID vaccine trial. You need much, much larger trials because the incidence of COVID that you're looking for might be too low, and 100 might never be able to pick up that incidence, right? So in a lopinavir-ritonavir trial, for example, the sample size was set at 160 because the trial investigator decided that that would give them 80% power to detect the difference between the two arms with a p-value of 0.05, right? To pick up a significant difference between the two arms, lopinavir, ritonavir, and placebo, they realized that they need 160 people in each arm of the trial, right? So before we do a trial, before we even get approval to do a trial, we do something called power and sample size calculation, right? That's when we hypothesize what the expected difference between the groups might be, what is the normal incidence of COVID or whatever in each group, how many new cases of COVID will we find in each arm of the trial, and how much power and how much statistical significance do we need, and then we put that in a formula. That's how we decide on the power and sample size. So I've given you a checklist on the key critical appraisal questions. If you were reading a randomized control trial and writing a report on it, these are the sorts of questions you should be asking, right? And also there's a critical appraisal worksheet in the Google Drive, which gives you more questions that you can ask in a randomized control trial. How about some questions?
All right, we've got a number of questions. I'll start off with the first one. Wouldn't it be more beneficial if you deliberately gave the vaccine to the obviously more exposed or at-risk group? This way, really testing the efficacy of the vaccine candidate.
Great question. So let's say we are most worried about elderly people, right? There is absolutely no problem in doing a vaccine trial only on elderly people. But even within elderly people, there will be one group that gets the vaccine and one group that will be assigned to placebo, right? Because we don't know whether the vaccine works. So we can limit the trial to the highest risk exposed groups, no problem. But even within that group, unless we have a treatment arm and a control arm, and unless it's done randomly, we will never be able to tell whether the vaccine works or not. Does that make sense?
Yes, it does. The second question, how serious can we take the first or second phase of the trial? In other words, at which point do we as journalists report on a vaccine or medication trial stating that there has been a breakthrough, for instance?
Oh yeah, great question. So for right now in COVID, every little trial is being cheered. Phase 1 is getting cheered, getting published in the New England Journal of Medicine. Phase 2 is getting cheered and reported and getting published in the New England Journal of Medicine. But in normal pre-COVID life, everybody would have gotten excited only with the phase 3 trial. Phase 3, double-blind, placebo-controlled, parallel arm, RCT, that's sufficiently powered is when you get super excited about it. And that's what we have with some of the treatments now, right? Hydroxychloroquine is practically dead right now because there's enough phase 3 trials which show that the drug doesn't work. And as of now,
Right.
there is no phase 3 COVID vaccine trial at all. They're all just starting. All we have is phase 1, phase 2 showing they're reasonably safe, right? Nothing terrible has happened. But none of them have anything on efficacy. All we have is that it's reasonably safe and it's immunogenic, right? Meaning it evokes antibodies. That's about all we know right now. Real rubber will hit the road with phase 3, which is where all of them are at now. Moderna, Oxford, everyone is entering phase 3. When that phase 3 comes out, that will be super duper headlines because it would tell us that the Moderna vaccine is 30% efficacious, 60% efficacious, 95% efficacious. That's when the real efficacy of any of these vaccine candidates will become known to us. Next question.
The next question is, what is the use of having trials where no one is blinded? And when we report on it, how do we state the shortcomings of these trials?
Well, you should always say that it's an open label, unblinded trial. So there's potential for information bias in terms of information collected or information volunteered by the patient. So it's worth mentioning that in your report. What is the point? Well, sometimes it's impossible to blind, really, right? You simply cannot blind someone for an intervention. So open label trial is the only practical alternative. Or sometimes it's faster and easier to do an open label trial. We would still consider it preliminary. We would still consider it inferior quality of evidence compared to a blinded trial if that's possible.
Okay. Shall I ask one more question and then we move on because of time?
Yep.
The last question is, how do researchers know that a vaccine was successful? For example, if say 3/4 from the test group remain COVID-free, is that a successful vaccine?
Again, depends on what the comparison group is, right? It's all relative. I don't want to only know whether people who got the vaccine, certain proportion remains COVID-free. I want to know what happens to the control group, because who knows, the control group may also have the same proportion of people who remain COVID-free, right? That's why vaccine efficacy is always set up as a comparison. A drug trial is always set up as a comparison. If I gave remdesivir only, I have no comparison group. And if I told you 50% of sick patients with COVID got better, you cannot attribute that to remdesivir because you haven't told me what happens to people who don't get remdesivir, right? Maybe 50% of them will also get better. Then remdesivir has done nothing, right? Remdesivir's efficacy is 0. So we always have to think in comparative terms. So vaccine efficacy inherently is a rate ratio, right? Or a risk ratio or an odds ratio. Okay. I'll move on.
Yes. Yes, thank you, Madhu.
All right. Now we've seen what randomized trials are, why they're so desired and powerful and important. But real world being what it is, there are many, many situations where we simply cannot do a randomized control trial, or it's not possible to do a randomized control trial, or in the early stages, we only do observational studies, and then we might do eventually a randomized control trial, right? So cohort study is one of the strongest observational design. It is the strongest observational design. So we put them at one rung underneath RCT in terms of strength of evidence. RCTs would be on top of the pyramid, the best evidence possible. Then failing that, we would accept a well-done cohort study as pretty powerful evidence. So what's a cohort study? So in the Roman legion army, a cohort was a bunch of soldiers who were pushed into the battle, right? They had cohorts, they had legions, they had all these army classifications, right? So cohort comes from the Roman army terminology. So we start with a population that's at risk. Then we measure exposures, whatever they are on, medicine, smoking, weight, height, whatever you want. And then we allow natural history to happen, right? Whatever happens, happens. And then we follow up that cohort to see how many people fall sick, right? Who gets the disease, who doesn't get the disease. We can compute a real incidence. Incidence is in number of new events that happen in the cohort. The denominator is the entire cohort that was followed up, or the person time contributed by all the cohort members, right? And then we can do incidence among those who smoked in the cohort divided by incidence among those who did not smoke in the cohort. And then we compare the two incidences, we get incidence rate ratio, an incidence risk ratio, and then we figure out whether the exposure and the outcome are associated with one another. But we don't decide who smokes, who takes a medicine, who exercises, who's on hormone replacement therapy, who uses condoms. None of that stuff applies. We just document the exposures and then we wait for the outcomes to happen. That's what a cohort study is. How long do we have to wait? Well, it depends on the disease, right? If it's a common disease, we don't have to wait for long. If it's a rare disease, sometimes we have to wait for decades before we collect enough outcomes to be able to figure this out, right? So that's what it is. We take a cohort, we measure their, they should not have disease when we start observing them, and then we classify them as exposed or unexposed, or exposed at various levels, right? Whatever the exposure factor is. And then we wait for disease events to pop up, right? And then we compute the incidence in the exposed group, incidence in the unexposed group. The ratio of the two, or the difference between the two, gives us the measure of association that we're looking for. You could have large cohort studies, small, you could have long duration, short duration, simple or massive, elaborate. We could look at multiple exposures and multiple outcomes. We can do all sorts of things. In fact, once you set up a really good solid cohort, and if you can consistently follow up people, it'll pay off over many, many years because of the richness of the data that's going to accumulate. Here are some really famous cohort studies, right? If we know anything at all today about heart disease and risk factors for heart disease, then we owe it to this community in Massachusetts. The community is outside Boston. It's called Framingham, a world famous cohort study called the Framingham Heart Study, right? Basically, they went, for whatever reason, they picked a small community called Framingham outside of Boston, and we said, would you all participate in the study? And this was in 1948, before cholesterol was proven to be a risk factor, before so many things we know today about heart disease was established. And they said, we will screen all of you for your blood sugar, cholesterol, your diet, your physical activity, your body mass index, everything. Come to a clinic, we'll screen you all. And then we will just wait and see who ends up with heart disease, stroke, whatnot, right? They started off in 1948, and take a look, that cohort study is still happening today, right? Here is 3 generations of participants, grandfather, father, son, all participants in the cohort study. So that cohort study is 70 plus years and still going as of today, right? They keep on collecting data. Every 2 years they go, they screen, they check everybody for their exposure and their outcomes. In Africa, I wanted to give you an example. This is the famous Rakai community cohort studies in Uganda, right? This was initiated in 1994, and it's an open population-based cohort. Enrolled all consulting adults between the age of 50 to 49 in some 50 communities. And this has really produced great data on HIV, AIDS, and sexually transmitted infections, right? Who is likely to get HIV, who is likely to have HIV and other diseases. Hundreds of papers have come out of the Rakai community cohort study, right? So like I said, we have cohorts from geographical region, by occupational health group. There's a very famous nurses health study where nurses have been enrolled in America, and the cohort study has been running for years. By disease, HIV study. By risks group, right? San Francisco Gay Men's Health Study was gay men in San Francisco that have been followed up for a long period of time. Or follow-up of all the Japanese atomic bomb survivors to see whether they end up with cancer later on in life, so on and so forth, to be based on an event. All the fire workers, New York Fire Department, who were exposed to the 9/11 terrorist attack, all of them inhaled massive amounts of smoke and dust, and they are still being followed up to see what their long-term lung capacity, lung cancer, so on and so forth, right? All examples of cohort study. You could begin with a cohort study in mind, plan for it. Requires a lot of money to be able to do that. Or you could do something like what I did. This was my PhD thesis work in India, where I surveyed, almost 700 healthcare workers, doctors, nurses for TB infection. And then 2 years later, I went back and I repeated the study on them to see how many people newly got infected in the 2-year period. Then my student went back many years later, contacted all the participants on the phone to see who developed TB or not. So we started off with a cross-sectional study. We never really planned to do a cohort study. But once you've done the cross-sectional study, if you can go back and recontact the same people and remeasure outcomes and exposure in them, then the cross-sectional study gets converted into a cohort study, right? So there's no magic. A cohort study is a series of multiple cross-sectional studies strung together to figure out who gets newly infected, who gets new disease, and so on and so forth. And how do we collect exposure information? Well, you could use occupational records, you could use interviews, you could use medical records, pharmacy records, vital statistics data, questionnaires, interviews, periodic health exams, blood sugar samples, blood pressure instruments. You can do, ECG. You can do all sorts of tests to collect information. So long as you repeat that kind of information over the same set of people, you get very rich data that you can analyze in the cohort study, right? Cohort study is of two types generally: prospective cohort study, retrospective cohort study, right? They are easy to understand. So let's say this is the, a year when some event happened, right? For example, 9/11, right? So you define the fire workers that got exposed in 9/11. They did not get randomized to exposure, right? It just happened. So some of them were exposed to smoke and fire and dust. Others were less exposed. And then you follow them over the next 20 years, for example, to see who gets lung cancer, who doesn't, the exposed group and the unexposed group. So you start with their exposure at the time it happened, and then you follow them over the next 10 years, 20 years, 5 years, whatever you want. That's prospective, right? So Framingham Heart Study is a prospective study that started in 1948 that is still happening right now. Right, here is an example of a prospective study on cohort study on COVID-19 that has already started. Big study by the Oxford University. Large multinational prospective cohort study with the aim of assessing the effect of COVID-19 on pregnancy, on maternal, fetal, and neonatal outcomes worldwide. They're going to already, this study is already begun. Okay, it's going to be a large study with over 500 exposed women and 5,000 non-exposed women. They're asking a very important question. If you're pregnant and you got the coronavirus infection, right? Will it have any bad consequences for the time you're ready to deliver the baby, or subsequent? Will the baby have any fetal malformations, for example? Is there any risk of bad outcomes for the mom or the baby afterwards, right? Really important question for which we really don't have much answers at this point. Sometimes we can do a retrospective cohort study, right? Somebody may have already collected a whole bunch of information in the past on a group. But we walk in now, today, for example, this could be 2020, we walk in now and check who has a disease or not. We're being opportunistic. right? Somebody's already documented the exposures for us in the past, and we go in now and we only look for outcomes among that group now. But we would still analyze it as if it's a regular prospective cohort study. Nothing changes except that this is a shortcut, right? We already have documented. So for example, you could go to the gold mines in South Africa, right? Let's say the gold mine employers have, smoking, asbestos, amount of time spent in the mines, other risk factors, alcohol, whatever, and they have documented it in their occupational records. We walk in now, we contact those people on whom data were collected in the gold mine, say 10 years ago. We check them now to see how many of them have developed, say, lung cancer or tuberculosis, and then we analyze the data, right? We didn't start the study 10 years ago, and then we didn't stick with that cohort. We just walked in now, but the exposure is already available for us from the past. That's called a retrospective cohort study. Here is an example. A retrospective multi-center cohort study of patients with confirmed COVID in 25 hospitals in New York City. The hydroxychloroquine or azithromycin was not randomly allocated at all, right? Some patients were already on it. Some doctors are already given it. Others had not given it. There were all sorts of people who were on the medicine, not on the medicine. You just collect the data from these 25 hospitals and you analyze them and you see whether hydroxychloroquine and azithromycin have any impact on mortality, for example, right? So exposure was being put on hydroxychloroquine or azithromycin or a combination of the two or neither, and primary outcome was in-hospital mortality. Did they live or die in the hospital, right? And that's the kind of data that they analyzed, right? So they found that the drug was not really helpful. If at all, the drug showed some tendency towards harm, right? The risk ratios were away from 1, but there was in fact greater odds of cardiac arrest in those who received hydroxychloroquine, right? And that's the kind of analysis that they would have done to look and to see whether the hydroxychloroquine group fared better. And they obviously would have adjusted for a lot of confounders in this analysis because the decision to put somebody on chloroquine was not random, right? So you must adjust for other covariates or confounders in this analysis. Cohort studies can also suffer from many biases. I think we've already discussed some of them. Confounding is a massive concern in a cohort study, right? And that is a concern we rarely ever have in a randomized controlled trial. I just told you this hydroxychloroquine observational study in New York. Confounding would have been the biggest issue they would have worried about, right? What else is different between the group that was given hydroxychloroquine compared to that wasn't given hydroxychloroquine? As compared to a randomized trial on hydroxychloroquine, where the decision is not in the patient's hands or the doctor's hands, it's randomly assigned, right? So that's the critical difference. Confounding is the single biggest concern that separates a cohort study from a randomized control trial. And it's not just for COVID-related stuff, right? We've known this to be true for a long, long time. This hormone replacement therapy is a classic textbook example of what happens if you only go with cohort studies and you fail to do randomized control trials, right? So they did all sorts of, so this is a very famous nurses' health study. Whole thousands of nurses in America were asked about whether they're taking HRT or not, hormones or not. And then they were followed up to see who gets cardiovascular disease like heart attack and stroke. And they found a strong effect. They said women who took HRT had a much lower risk of heart disease, right? Large, huge cohort studies, many such cohort studies, and they all seem to suggest that hormone replacement therapy really protects against heart disease. And then it became widespread practice, right? Women were asked to take HRT. I still remember that in the early, late 1990s, lots of women were under great pressure to start taking hormone replacement therapy to protect against heart disease, right? And yes, there were cohort studies. And yes, they did adjust for confounding. See, that's the original cohort study paper. You see, they adjusted for age and risk factors, a whole bunch of risk factors they adjusted for, right? So they were worried about confounding because they thought women who took HRT would be very different from women who did not take HRT, and they tried to adjust for it, right? They adjusted for cigarette smoking, they adjusted for diabetes, they adjusted for age, they adjusted for past use of contraceptives, whole bunch of stuff, right? Even after adjustment, these cohort studies still saw that taking hormone replacement therapy was protecting women from heart disease. That was the understanding at that time. And then the massive first randomized control trial gets done. Very famous trial, the Hearst trial by the UC San Francisco group, right? And they found, to their horror, that hormone replacement therapy increased the risk of heart disease rather than decrease, right? Complete flip in our understanding of the effect of hormone replacement therapy. Totally changed the field in terms of how we would approach hormone replacement therapy, right? And then confounding was absolutely not a concern in the Hearst trial because it was randomization that worked. If you look at the group that got hormones and the group that got placebo, you see a beautiful difference, right? If you thought age was an issue, look at that. 67 years in the hormone group, 67 years in the placebo group. White women, 88% in the hormone group, 90% in the control group. Education, 13 years of education, 13 years of education, right? Smoker, 13%, 13%. It's ridiculous. It's almost like somebody fudged this, right? That's the power of randomization. If you randomize large enough people, you will get spectacularly comparable groups. So none of these variables are confounders because you made sure that the two arms of the trial, the hormone and the placebo, have very, very similar distribution of the confounders. Smoking, I don't care. Diabetes, I don't care. Body mass index, I don't care. Do they exercise more? I don't care, because no matter how I look at it, they are similar in the two arms of the trial. That's why we would believe this Hearst trial, even though there were many cohort studies showing that hormone replacement therapy was protective, right? So again, I've given you a list of critical questions to ask if you were reading a cohort study and you were writing a report on on that paper. And there's obviously a more detailed critical appraisal worksheet in your Google Drive as well. Okay, questions?
Hi, Madhu. So the first question that we had in this section is, what is the best design for a cohort study, qualitative or quantitative? I thought we'd address this one again because you mentioned it in the last session, but it's come up again.
So everything in epidemiology is quantitative, right? We do not do qualitative research in epidemiology. So this entire course is quantitative. There is no qualitative research at all in anything that I've taught you. So if you say a cohort study, it's quantitative.
All right. So here we have two questions that are interlinked. And the first one is, wasn't it too early to write off hydroxychloroquine? What will happen to cohort studies that might be done to assess efficacy of hydroxychloroquine in Uganda? So I think the backstory is that there are still trials that may be ongoing for hydroxychloroquine in Uganda. And then a linking question to answer the same thing is, if another trial of the same drug therapy is done and concludes a completely opposite finding, does it immediately render the first one ineffective?
Great question. So there are now, I think, 4 or 5 published randomized trials on hydroxychloroquine. And we can try and combine all of the published RCTs on a given topic through a design called meta-analysis, right? We could do a systematic review. search for all the trials on hydroxychloroquine, put them in one place. And then if certain criteria are met, we can mathematically combine the trials and compute a pooled risk ratio. Karen Young will show you or teach you more about that next week. In fact, on Thursday, the last class. So a meta-analysis is still helpful because there may be multiple studies and we want to synthesize all that evidence in one place. Could we have trials that disagree with each other? Absolutely. Has that happened? Yes. How do we then break a tie? Well, we critically look at the quality of each trial, right? One might be a very well-designed, well-conducted study. The other might be a really badly done trial. So in which case, we will weigh or emphasize a well-done trial over a poorly done trial, right? So just because it's a trial doesn't mean that it's done well or done correctly, right? Number two, much larger trials, mega trials, are often weighted more because they can be very conclusive, they can be very large. But mega trials could be done badly as well. So we need to look at the quality and we look at the totality of evidence. If we have 8 randomized trials on hydroxychloroquine and 7 of them show that hydroxychloroquine doesn't work, I think by and large, we would be swayed by the amount of evidence that we are seeing in that. And the meta-analysis will tell you how to understand that. All right.
Yes, thank you, Madhu. That's all the questions we have for now.
All right, great. So now the last mini lecture is on case control studies. So think about a pyramid of evidence where the best evidence is typically a randomized control trial. Then comes a cohort study. A well-done cohort study is very important. And then we also would say a well-done case control study would be maybe a little lower than a cohort study, but a well-done case control study is actually pretty much as good as a cohort study. So here is an example of a case control study, right? Acute onset smell and taste disorders and COVID-19, right? They wanted to know, is there a link between smell and taste disorders and having COVID? So they took as cases 79 consecutive patients positive for COVID-19 using PCR. And they took 40 patients from the same hospitals who didn't have COVID, but they had influenza, okay, some other disease. So cases are COVID cases. Controls are non-COVID people. In this case, controls had influenza, okay? And then in each group, they asked, do you have a problem in smelling? Do you have problems in tasting your food and so on and so forth, okay? And then they put the results together into this simple 2x2 table, right? Cases, the first column, controls, right? People with smell and taste disorder, no smell and taste disorder, right? And if you did the math, as you've heard from Ngozi's lectures, we can compute something called odds ratio. Odds of smell and taste disorder among the cases divided by odds of smell and taste disorder if you are a control, and you do the number crunching, you get an odds ratio of almost 5. What that means is cases, COVID cases, had a 5-fold higher odds of having smell and taste disorder as compared to the controls, in this case, influenza, right? COVID patients compared to influenza patient had a 5 times higher risk of having smell and taste disorder. In fact, we now accept that as a widespread, well-known fact that COVID can lead to anosmia or not able to smell. It's a very peculiar symptom of COVID-19. Here is an example of a case control study on hydroxychloroquine for prevention of COVID-19. This is from India, right? So they had a whole bunch of healthcare workers. Some of them had taken hydroxychloroquine, others had not, okay? So nothing was randomly allocated. So they found patients who ended up with COVID. Then they took up healthcare workers who did not have COVID, and then they asked who was on hydroxychloroquine in each of the 2 groups, right? And that's how they compute an odds ratio to look at the association between COVID and hydroxychloroquine. right? So the way we think about the cohort case control study is almost as if it was a smart shortcut to doing a cohort study, right? So let's look at a cohort study, okay? Let's say this is the Framingham cohort study, okay? Whole bunch of people were asked about exposures to various things, diet, physical activity, blood pressure, so on and so forth. And then they were followed up over time to see who develops the disease. The black dots are all people who develop the heart disease, right? This is a normal cohort. You start at the baseline, you measure the exposure, then you wait until you have disease or illness popping up in the cohort, right? So those are the cases that would occur in a cohort. And we would get something called the incidence. Incidence is the number of new events that occur in the total denominator, or the number of people that are being followed up, right? But what we could do is, instead of doing this long, painful waiting, we could start off with people who develop heart disease, okay, in a region, for example, Framingham, right? We get all the black dots and we recruit them into the study as cases. And then we need to get recruit a group of people that we call controls. Now, who is the best control? The control is someone who is available at the same time as the case event happened in the same population, okay? They could have become a black dot, but they didn't. They could have developed heart disease, but for whatever reason, at that time, they didn't. They could become a case later on, we don't know, okay? But at the time a case was recruited into the study, we should get a control who does not have the disease, but could have gotten the disease, but hasn't, right? If we could do that, now you see there are plenty of potential controls, right? There are literally dozens of people who could have become a case, but they didn't, right? What we could do is, for each black dot, we could at least get one matched case control, and we recruit them as pairs. That's called as a matched pair case control study, right? So if we have the cases who develop the illness, and then we have a bunch of people who could have developed the illness in the same population at the same time, but they didn't. And we take a random sample of the controls or a matched sample of the controls, then we have a case control study. Now, if we did that, we would then get something called an odds ratio. And the odds ratio we get would be almost the same as a risk ratio we would have gotten if we had done the whole cohort study all by ourselves. And that whole cohort study would have been time consuming and expensive, and prolonged time period may have been required. We kind of shortcut all that by doing the smart sampling of cases and a smart sampling of controls. So case control study is a very smart, efficient way of getting the same information that you would get in a well-conducted cohort study, okay? Powerful design in epidemiology. In fact, epidemiologists pride themselves on this design because it has saved us endless headache and money in doing cohort studies, right? So if we can do a cohort case control study to answer a question in epidemiology, we'll always do a case control study. It's smart, it's efficient, gets us the answer much quicker, much cheaper, less expensive than a cohort study, okay? Now, question is, who are the controls that we could recruit? There are plenty of different controls, okay? We could recruit controls from the population. We could recruit them from the same hospital from which cases were diagnosed. We call them hospital controls. We could recruit the family members, right? So if you recruit someone in the family with COVID and the remaining family members don't have COVID, you could recruit a family member who doesn't have COVID, right? You could recruit a friend. You could ask for a list of your friends, the case's friends, and recruit someone randomly from that list. So there's also random digit telephone dialing. You can dial random digits in the telephone and recruit someone from the population or the same district, and so on and so forth. And each one has its own limitations. The ideal control would be the population control if you could do it well. But in practice, it's hard to do So you will see many case control studies where the controls are hospital controls, right? They're usually from the same clinic or hospitals where cases were found. Selection bias is a huge issue in a case control study. The biggest question we ask whenever I read a case control study, the first question I ask is, how the hell did they select the controls? Nothing is more important for me than how controls were selected. Because if I get a wrong or a bad control group, I can pretty much show you any result that you want to see. Okay, so the control group should be from the same population from which cases came, and they could have become cases, but they didn't. And you need to do it in a random or a clear pattern rather than cherry pick those who you want. Okay. And then there's also information bias. So let me give you a quick example of how a biased control group can skew the study results depending on how you look at it. So this is a famous case control study by my boss. He's an epidemiologist who trained at the US CDC. And so in those days, in the 1980s, there was a lot of anxiety about something called a toxic shock syndrome, right? The men, after having their periods or during their periods, would suddenly go into septicemia and shock and die. And he was investigating what was the reason why these women were having this toxic shock syndrome. And so he did a case control study. He recruited women who had toxic shock syndrome during menstruation. And then he recruited controls. He recruited two types of controls. They were all matched on age. They were all women, obviously, so sex matched. One controls were friends of the case. So he asked the women, give me a list of friends of the same age group as you are in. And one of them will ask to be a control. And then they also did a second control group of telephone exchange. They randomly dialed from the same telephone code women until they found a woman of the same age, and then they invited them to be a control, right? Now take a look at the results in terms of what they found, right? So they found that use of tampons, okay, tampons in the end was one of the biggest risk factor in this study. And we know that to be true. Tampons that were soaked with blood ended up getting contaminated, and that caused septicemia, right? So if you look at the friend controls, the odds ratio is 19. When you look at the neighborhood controls through telephone digit dialing, the odds ratio is almost 50, right? Big difference between the two odds ratio. Why is that? They found in the end that friend controls was biased because of this, right? So this is a 2x2 table, right? The cases and the controls, by selecting friends of the cases, you kind of made sure that the cases and controls were more alike, right? Now, if I'm of a certain age, if I'm going out smoking or drinking, or if I abuse substances, is it likely that my friends are also doing the same thing? The answer is yes, right? So if I'm a young woman and if I'm using tampons, do I also have friends who might be using that? The answer is yes. So friends generally tend to have similar risk traits or behavioral traits, or they may be exposed to the same type of activities. If I'm physically active, I go biking every day. Is it possible that my friend also goes biking with me? The answer is yes. So you make the cases and controls more and more alike, and then you inadvertently end up overmatching them, right? So this is a problem. That's why the odds ratio was much lower than the real odds ratio, which they got from the community, right? So this is a nice example of selection bias. If you get the wrong control group, it can completely skew your results. And I also gave you a similar example last time of coffee and pancreatic cancer, right? If you have cases who have pancreatic cancer, and if your controls are people with other GI disorders, and they've all been told to stop drinking coffee, that skews the whole odds ratio and made coffee look like a big risk factor for pancreatic cancer when they actually are not associated with each other, right? All right. So information bias is another big concern. There is poor recall, right? In a case control study, we often interview the cases and the controls, and we ask them, so what did you do, right? What diet do you take? Who are you exposed for? Did you smoke in the past? All sorts of questions, right? So sometimes poor recall is a big problem. Poor recall is something that everybody is suffering from, right? Even I don't remember what I ate a week ago, right? So poor recall is always a given. But recall bias is a unique problem in a case control study where cases recall differently from the controls, right? And remember the discussion we had last time. If I'm a mom who just gave birth to a baby who has a major congenital problem, then my recall of what exposures I had during pregnancy will be very different if I just gave birth to a healthy baby, right? Cases and controls will remember differently, will recollect differently, will ruminate differently, and that can completely skew your study results, right? So this is a big issue. And there are all sorts of interesting biases. Case control studies are prone to serious biases. Here is the spectacularly biased case control study that is published, which I use a lot for my teaching example, right? So here is a study on risk factors for committing suicide in Pakistan, okay? Pakistan. So they found, here are 100 cases, here are 100 controls. They found a spectacular levels of depression in the cases, right? So 30% moderate depression, 43% severe depression. If you add up everything, pretty much everybody in the case group is depressed and pretty much nobody in the control group is depressed. Okay? So, and you end up with a ridiculous odds ratio of 208 for depression. This itself should have made these authors slow down and say, oh my God, what kind of an odds ratio are we getting? I mean, how could you have a control group that is completely kind of sanitized? Nobody has any mental health issues. Pretty much every case has a mental health issue. And then if you turn and look deep into the study, you realize where the bias is coming from. The cases are all dead people. They've all committed suicide. So then the only way you will find any information about them is interviewing the next of family, right? Now, Pakistan is a conservative society, just like India. Suicides are rarely discussed in public, right? Family will not talk about that to anyone, right? So when they interviewed the next of family, everybody must have said, yes, my son, my father, my daughter, they were depressed. They committed suicide, right? So everybody apparently had depression. Now, controls were people living in the same neighborhood or street as the person who committed suicide. And then you find someone who's living, and then you ask them, right? And lo and behold, nobody has depression, right? So the way the information was collected completely changed. So nobody was blinded, right? Everybody knew what the study was all about. And the quality of the interviews might have been very low in the controls, right? In the controls, I know you're a control. I know you are alive. You didn't commit suicide. So my probing of your mental illness might be much less. On the other hand, I'm interviewing the next of kin of a dead person who I know committed suicide. I can probe very hard and make sure I find out whether they're mental illness or not. And I see a massive difference between the two arms. Spectacularly biased study that should not have been published, in my opinion, but it's published in the British Journal of Psychiatry. All right, so confounding is a last issue. Always worry about confounding. Like in all observation studies, we'll match for to adjust for confounding. We could do multivariable analysis, logistic regression, and we will generally report not the crude odds ratio, but the adjusted odds ratio. Odds ratio adjusted for age, sex, socioeconomic status, mental illness, whatever you want to adjust for in the analysis, right? So confounding is something we would always be concerned about in a case control study. Time for a couple of quick questions before we close.
There were no questions in this section, but perhaps I could go back to one question that came up twice in the first section about randomized control trials. Mm-hmm. And the question was, how do you are there a minimum number of arms to be used in a randomized controlled trial? And then the follow-up question was why in RCTs individuals must receive at least 2 interventions, not 3 or 4 or 1. So I think instead of interventions, what the person meant was why are there 2 arms in RCTs?
Well, we need to compare a drug against something, right? Or a vaccine against something. So two arms are typical. But if you want to do 3, you could, right? So you could have a randomized controlled trial where one arm gets chloroquine, another arm gets remdesivir, third arm gets dexamethasone, for example, right? And a fourth arm could get placebo. It's all up to the investigators on how complicated they want to make it. But at a minimum, you need a treatment and a control arm. The control arm could be a placebo, or the control arm could be some other drug, right? So a 2-arm parallel RCT is the most basic type of RCT that can actually produce valid results. A single-arm trial will never really tell us whether the drug works or not. Single-arm trials are more helpful for safety, side effects, and so on and so forth. Any other?
Thank you, Madhu. That's all the.
Yeah.
No, there are no more questions.
Anyone
Thank
wants
you.
to actually speak and ask a question? You can unmute yourself and ask as well. You can turn on the video and unmute yourself if you want to ask a question. Anyone? All right, great. So thanks everyone. We will see you for the last lecture on Thursday. We need 2 hours of your time to close out on all the key topics we want to cover. Rosalind will follow up after the last class with an evaluation, quick survey. Please give us your feedback. Taryn might be thinking about a similar course for African journalists as a follow-up. So your feedback would be very helpful for her. And then we will also, Chido and Ia will also share with you the post-course quiz after we finish the last lecture, so that you can check yourself how much you've improved in your understanding of basic concepts in epidemiology, okay? And as always, I will post this video and the slides are already posted as well. All right, have a good night and please stay safe. Bye.
Thank you, Prof.
Thanks, Madhu. Bye, everybody.